Okay, let's get started. Okay, are we ready? We are? It's Friday. There's so much excitement in the room. And it's such an exciting topic today. Or not. Okay, we'll live through it. Okay, so we barely started talking about sampling and sample preparation last time. Started with a couple of questions, so not much yet for a recap question. We are starting with a population, sampling from a population. That means the entire lot, either production in a week or the truckload of products or whatever. This is your population. And when you have the entire population, we asked, can we use the entire population for sampling? Is it possible to use 100 tons of grains to take them all and sample them? No. So unless it's a rapid analysis, like just screening for metal, goes through metal detector or color or sorting. So non-destructive and kind of a rapid procedure, you might be testing the entire population. However, oftentimes, or most of the time, 99% of the time, you have to take a sample. Actually multiple samples. So sampling from the population, and it's very important, so when you're asked to talk about this, when you say sampling, you have to remember it should be representative of the population. So whatever sample you picked, whatever the results of the analysis is, your decision is made on the whole population. So in order to determine the fate of the population, the samples you withdraw from the population, the results should be representative for the entire population. And in order to make sound decisions, we have to have accurate estimation to reduce variance. So where your variation between analysis is reduced and your values are trustworthy, you're more than 90% sure of your results. Because big decisions are made, either sometimes it's reject or accept a lot. So there would be consumer risk and there will be the industry risk. So the risk to the consumer is to accept something that has poor quality that might cause them sickness or illness. The consumer to the vendor or the producer is you reject a lot that probably is good, so they would lose money. So that's why it's really important that you take a representative sample of the procedure in order to get accurate estimation and sound decisions. So in order to do that, you have to come up with a sampling plan. And it's a very common question I would ask in a quiz or an exam. Define a sampling plan. What is a sampling plan? What does it constitute? It constitutes several things. First of all, what is the goal? So in your sampling plan, you have to identify the goal of your analysis. What are we analyzing for? I'll show you some questions that you would ask for the goal. So stating the goal is one thing. Determining sample size, how many samples. Again, the size here means how many samples rather than amounts is the meaning of sampling size here. How many samples I should get? Sometimes you're bound to regulatory. If it's nutrition label, we'll talk about that. You're bound to a certain amount of samples that you have to pull. Other times it depends if you're doing attribute sampling or you're doing variable sampling. I will talk about that. So really sampling size, sampling procedure. So how are you going to pull out a sample? Okay, we'll talk about sampling procedure as well. And then sample storage. We'll talk about sample storage and sample preparation. Sometimes you pull out sample and they have to wait for a certain period of time until you're ready to process them, ready to prepare them for analysis. And after you prepare them for analysis, sometimes you also have a holding time before you actually run your samples or analyze your samples. So this all goes into your sampling plan. So when you see sample plan, I say define. Now you should be able to define what a sampling plan is. Okay, so factors to consider when you're designing your sampling plan, and that was a big part of your project, if you were to do a project, is to design a sampling plan at each analysis point. So what would you ask to determine your sampling plan is, again, cause. What is the purpose of the inspection? Is it to accept or reject a lot? So are we looking for Clostridium botulinum in our canned food product? So is it, if you have Clostridium botulinum, you're going to reject the lot. So there is zero tolerance for that. So is it to accept or reject the lot? That's a big question. Is it to measure quality of the lot? We're looking at sensory acceptability, for example, or we're looking at composition, variability between different production of different days. Are we getting the same composition? Are we getting the same consistency? Is that the purpose of our sampling or plan? What is our products, the nature of the product? Is it homogeneous? Sampling is easy. When you have a homogeneous sample, then anywhere you pick a sample is going to be the same. If it's a flour, usually it's not that hard to decide where to get a sample from a flour bag. It's homogeneous, it's flour, it's been milled. However, if it's heterogeneous, it's a non-processed food product, let's say, and you have a layer, a tomato, not tomato, let's say the apple or oranges, so you have the layer. It's not a heterogeneous sample, or it's a bar. It's not a heterogeneous sample. What do you need to do to make it homogeneous first before getting a sample? How big is your unit size? Are we looking at cans? Are we looking at flour bags of 40 pounds? What are we looking at? And that would determine how many samples and how many samples from each container. Is it a box of 100 cans? What are we looking at in terms of unit? How consistently have past population met specification? If past populations have almost, your production have almost passed, then you might not be sampling a lot of samples. You know that by experience that you probably shouldn't be sampling 100 different containers, let's say. You might go, I'm pretty sure I've met specification most often, then I will reduce the amount of sampling that I would do, or the number of samples I would pull out. What is the cost of the material being sampled? That is something you would consider. Can you afford taking that many samples and testing them? Because you're basically, most of the analysis is destructive. So you take the sample and it gets destroyed. So then you have to think, how expensive is my sample? And take that into account when deciding on sample size. So what type of analysis are we doing? Is it a critical test or a minor test? Critical becomes, is someone going to get sick? Are we looking for mycotoxins, for example? How many samples we should gather? It's not homogeneously distributed. Fungus can grow anywhere on your sample, and it could be, and the fungus produce mycotoxins. So you could have mycotoxin in one container and you might not have it in another. So you really have to increase the number of samples because the risk is high on the consumers. And then you consider, is it a destructive, non-destructive? Usually when it's non-destructive, it's easy. You can do a lot of different samples. You don't care because you're not destroying your samples. You can do multiple analyses after that. And then how much does the test cost? How expensive is the testing method? Nature of the population being investigated. How big is the lot? The lot is large but uniform. So container of wheat, they're all wheat. Or flour, it's uniform, large. We can take samples, limited amount of samples because it's mostly uniform. Does the lot consist of smaller, easily identified supplements like cans of food? What's the distribution of the units within the population? Are the units packed in boxes and how many boxes and how many cans within a box. So all of that is things to consider in determining the number of samples and the location of samples that you would get out. So there are two types of sampling. There's attribute sampling and variable sampling. And often students confuse the two. I sometimes forget what the difference is. But attribute sampling is usually when we are looking at a particular attribute that is randomly distributed. That means it's not homogeneously distributed within your sample. For example, if you have flour, protein is homogeneously distributed in your sample. It's a composition. Whereas if you have fungal growth, it might not be, or mold, it might not be homogeneously distributed. So it would be looking for a particular attribute that is not uniformly spread within your sample. So randomly distributed attribute. In this case, in order to capture the presence of an attribute, you would really need to take many samples. So your sample size is high. That means the number of samples that you select is high because you want to make sure you capture if it's present. If you only decide to take 3 samples, that's a limited number of samples. You might not get a sample that is contaminated. So in attribute sampling, key is large sample size. And it's often checked for presence or absence of a variable. And then you decide acceptability, reject or accept a product. And here's an example. Clostridium botulinum is one. One example for there is 0 tolerance for its presence. So Clostridium, you have 0 tolerance, but other organisms and other components could have an acceptable level of presence. So you have 2 choices. If it's present, you have 2 classes. It's there, positive. So either you would go no, that means the count is 0. There's no tolerance for anything. So if it's present, then you would reject. If it's present, then you go and do the count and determine if the count is greater than 0, and then you would determine if it's within acceptable level. If it's within acceptable count, then you would accept it. If it's bigger than the acceptable count, then you would reject it. Variable testing is easier. It is, like I said, for uniformly distributed attributes, like composition is uniformly distributed attributes. So protein, fat, moisture usually is uniformly distributed within the sample, unless it's a sample that is not homogeneous, it's heterogeneous, but you would mill it first, make it homogeneous, and then from there you will get less amount of samples because it is homogeneous from there on. So 2 things you have to keep in mind. Is your sample homogeneous, heterogeneous? Are we looking for variables or are we looking for attribute? So sample size is smaller when we have normally distributed attributes or components. And oftentimes it's quantitative, it's not qualitative, it's not just present or absent. So you are actually detecting composition or quantity on a continuous scale. And then you decide acceptability. Does that final value is it within your specification? Is it within your standards? Then you make a decision on that. And often you compare to an acceptable value that you've been working with or regulatory recommends. So here's an example of what we're measuring: color, total solids, salt, and other compositional components. So selection of sample size depends on so many factors, oftentimes overall quality and sampling cost, but in some cases you are bound to regulation, especially for nutrition label. So if it is for nutritional label, FDA and FSIS have regulations, and these regulations stipulate that you need at least 12 samples. So you need to gather 12 samples, and we'll talk about randomly selecting your samples in a minute. And then from these 12 samples, you form a composite, and that's the regulation. You get 12 samples, and then you mix them together, homogenize them together, form a composite sample. And from that composite sample, you take 6 replications, subsample. That is the requirement by FDA and FSIS. Okay. So we have 4 different types of acceptance sampling. Acceptance sampling meaning when you sample, you get results, and then you decide whether you accept or you reject your lot. So there is something called the single accept or reject based on one set of samples. So you would determine how many samples you were going to get. N is your sample size. And then C is the number of defects. So for every type of analysis, there is a C, number of defects. Sometimes you set that C as a vendor or producer. Sometimes you are bound by regulation. You set the C, or the C is set by regulations. So you would just take one set of samples, and whatever is the C, you will make a decision either to accept or reject based on the C that you've gotten from one set of samples. Double, here's an example of double accept or reject. So in this case, you have your population. You take your first set of samples. That means, let's say, 10 samples you decided to take. And then the number of defects, D stands for number of defects. If a number of defects is less than a particular standard that you have or a code, then you would accept the lot and move on. If the number of defects is greater than a particular number of defects that you set for yourself or by regulation, then you would reject. So this is easy. Lower than a certain number or higher than a certain number, you reject. But if you get number of defects in between, then you go ahead and take another set of samples. And that's why it's called double. At this point, you're taking a second set of samples with a set number of samples, that means sample size, and then you take the measurement. And then D1 plus D2, if your defects 1 and the defect 2 is less than another code that you have, you accept. If it's greater, then you reject. And like I said, these numbers, the number of defects that you usually compare to is set by the vendor. You have certain quality criteria or quota, or it's set by regulations. Now, the multiple one is you start sampling, and then you count the defects. And based on the number of defects you get, you either stop sampling and reject. Let's say you took 5 samples and you found 6 defects. That's it. You stop sampling and you reject. If you got 10 samples and you got 3 defects, you're still here. You continue your sampling. If you got, let's say, 40 samples and then you got 1 defect, you accept and you stop sampling. Okay? Time for a question. When monitoring food safety by measurement of mycotoxins, so you're measuring mycotoxins, many randomly selected samples are required. A single sampling plan is required. Sampling error is many times greater than the analytical error. Come on. Get excited with me. Yes, great. So B? I'm going to tell you hold the thought for now. Any other? Ted? A and C. Alex, where are you going to say? Okay, we had a variation of answers here. Okay, so mycotoxin, do you think it is uniform, would be uniformly distributed in your sample or not? No. So it could be randomly present where the fungal growth happened. You might have produced, the fungus might have produced mycotoxin in a particular region. So you really, when you test, you want to make sure that you covered every region and you took a lot of different samples because mycotoxins, some of the mycotoxins you have zero tolerance. No mycotoxins are allowed, especially mycotoxin B, for example. Aflatoxin B, I want to say, as an example. So you want to take many randomly selected samples in order to ensure that you don't have mycotoxin. B doesn't really work in this case because you might get a set of samples and if you, unless, okay, so this is here where it is kind of how, depends how you read it. Unless you take from the first set of sample size, you take a lot of samples. If you do that, you cover from multiple regions that one set of sample and then you found, then you would reject right away. So you are partially right, but I did not intend that to be the way you interpret it. But if you interpret it the way you did, then you're right. But what I was looking for is A and C. Sampling error is many times greater than analytical error. When you determine mycotoxin, the analytical method is well verified, and if you're trained on it, you should have good results. But if you make error in determining where to take your samples and how many samples to take, then you are more at risk of accepting a contaminated sample than the error that comes with the analytical method. Okay. So we talked about risks associated with accepting or rejecting a sample. We talked about consumer risk. And usually consumer risk is big, and we need to take it into account because somebody might get sick, and you don't want that. There will be legal matters associated with that. So the probability of accepting a poor quality population should be less than 5%. Zach?
Was the previous answer to the last question, was it just A or was it A and C?
A and C.
It was A and C.
Yeah. So the probability of accepting poor quality population should be less than 5%. That means you should be more than 95% confident with your decision when you accept a population. Okay. However, the vendor risk, you have some leeway. Of course, it's important because it's associated with profit. When you reject a lot, that means you're losing money. So you really have to be confident that you made the right decision, but here you have more leeway. You give yourself a little higher percentage of a mistake. So you go, okay, I'm going to be 90% sure that my decision is correct in rejecting a good quality population. In this case, when you reject, the risk is on you losing money. Here, when you accept, the risk is on the consumer getting sick. Or not necessarily sick, getting a product that is not of good quality, then they won't buy your product again. So to sum it up in terms of sampling plan, while you have to consider so many different factors, you might want to also, from a vendor perspective, you want to make it simple and flexible. You don't want to complicate your life. You don't have time to do a lot of samples or tedious analysis. You want to protect consumers and vendors, so the same sampling plan should take that into consideration, both the risk to consumers and the risk to vendors. And you want to ensure that your results are sound, the reliability of your results. So that's the sampling plan in summary. While you consider so many factors, you want to make sure that it has these components or elements considered. Here's an example of sampling of a flour. So AOAC have a procedure for sampling the flour. And let's say, first of all, for simplicity, you produced 100 sacks of flour. Each sack is 40 pounds, 100 pounds, whatever is the sack. And you want to select, not all of them, you want to select a number of these sacks for testing. The rule here is you take the square root of the number of the sacks in the lot. So if you have 100, you select 10. And then we're going to talk about random selection in a minute. How do you randomly select these 10 bags or sacks? Once you select those 10, you want to get a sample from each bag. So oftentimes you take 2 samples, drawing a sample from a corner diagonal to the center. So you would go, okay, I'm going to take from this corner diagonal to the center, and then take another sample diagonal to the center too. So you go from this corner. I'm going to take from this corner and this corner, or this corner and this corner. And then your samples will be stored in clean, dry, well-sealed container until further analysis. That is the methodology for, an example methodology for sacks of flour.
Do you combine, once you take 2 samples, do you combine them
You keep them separate in this case. Yeah. A sample for, if it's a homogeneous lot, then the sample can be taken from any location. No problem with homogeneous. Now when you have heterogeneous, you want to make sure that you get a sample drawn from multiple locations. So you can get manual sampling, which is prone to human error, or you can have mechanical sampling, which is oftentimes continuous and is less biased. And this is often performed with systematic, what we call systematic probability of sampling, or sampling probability. So we'll talk about that in a minute. So that means you, after, it's timed, like maybe after 2 hours or every 2 hours, the machine automatically selects a sample, picks up a sample, whether it's a can or something that you would, liquid soup, you would get it in a container before it gets canned. So that would be mechanical sampling. It's continuous, it's less biased, less prone to error, that is. Okay, so now let's talk about sampling procedure. Once you determined where you're going to get the sample, how many samples, then you will have 2 types of procedures, either non-probability, not based on statistics, or probability sampling. So non-probability is not often, it's done only when you have certain situation. Restricted sampling, that means, let's say you have a truck load and you have many boxes, so you cannot reach the box that is way inside the truck and at the bottom. So you are really restricted, so you're going to grab whatever you are able to reach to take for the testing. So that's restricted sampling. Judgment sampling is basically done by somebody that is have experience. So you have this truck of grain and it's been emptied, so there's somebody that comes and from experience grabs a couple of samples for analysis. Now, what is better is the sampling based on probability and statistics. So we call it probability sampling, and there are so many different kinds. One is random sampling. Have you ever used random number table? Okay, so some of you might remember that. So usually you would have a table with random numbers, not in any sequential order. And what you will have is you would take your lot and you would give them numbers. The units within your lot, you would give them random numbers that match the random numbers in that table. And then you randomly, close your eyes and randomly select, whether if it's 10, randomly select 10 numbers and go pick up those particular samples that were given the same numbers. So that is random sampling. Systematic sampling, we just talked about it, that is automated, and every certain period of time during processing, a sample is collected. There's stratified sampling. Let's say you have a company that has multiple branches, but they're all producing a certain like canned food, tomato soup, tomato paste, whatever it is. What you would do at this point is you would stratify. That means you will divide based on location, and within each location you apply random sampling to get samples from each location. Cluster sampling, in this case, you don't select, you don't test in all facilities. It's the same example. You have tomato soup produced in multiple locations, but all are produced the same way. So you would randomize these locations and randomly select a couple of locations for testing. You don't necessarily test everything. But again, here to decide on cluster sampling versus stratified sampling, it really depends on what you're testing for. If you're testing for quality and acceptability and you are having the same process in all of the plants, then you can decide, I'm going to do cluster. But if you're looking for Clostridium botulinum, you might have an outbreak in one plant but not in another, so you have to do stratified sampling. Make sure that you're testing all your plants' facilities. So really it depends on the purpose to select either stratified or cluster sampling. Last one is composite sampling, and that's the example I gave you when you have the nutritional labeling or composition for nutritional labeling, and that's what the FDA and FSIS dictates, is that to get 12 samples and then combine them and then do sub-replications, 6 sub-replications. So that's an example of composite sampling. But that doesn't only apply to FDA and FSIS for nutrition labeling. Other people might choose to do the same way, get multiple samples, homogenize them, and then do subsampling. So some of the problems, potential problems associated with sampling may result in reduced reliability. Which of these may result in reduced reliability or trusting of your data? Riley? Yeah, all of the above. So when you really have a restricted access to samples and you grab a box for convenience, then at the top, that is not a statistical way of sampling, then that's prone to error. Poor storage conditions might change and alter the sample for you, so you will end up having non-representative results of the lot. And then mislabeling, that's a common and obvious mistake. So in order to run the analysis, you really have to bring the particle size down to increase the surface area, because a lot of the analytical methods require higher surface area. So one of the first steps you would do, and you're going to experience that in the lab if you're going to work with a cereal, total cereal from General Mills, and you want to analyze protein content and fat content. So you have to grind that into a certain mesh number. So that you will experience in the lab, that you are reducing the particle size. So if your sample is dry and low fat, it's very easy to grind it. There won't be any problem. But if your sample has a high moisture content or is sticky like a nutrition bar or protein bar, or it has high fat like cheese. So in order to grind it, oftentimes we do what is called cryo-grinding. That means you grind it when it's frozen or with dry ice. So this way you will eliminate the loss of moisture when you grind and the stickiness problems, and also loss of like fat sticking on the sides, and then you will have an underestimation of fat and moisture. And there are some errors that would be introduced when grinding the sample, depending on what grinder you use. One of them is metal contamination, the other one overheating. When you have that, you have to make sure you don't get overheating because you lose moisture and then you underestimate moisture. You don't want to introduce metal because if you're determining iron, for example, you will probably overestimate iron. One time, it's a capstone group, I walked in, they were doing fat extraction using Muginer, and this method you're going to learn. And their product was potato chips, and there is special container for the Muginer, special flask. And I walked into the lab, and they were happily shaking and extracting, and I looked in there, and I saw a whole piece of potato chip in there. I was like, what are you doing? Oh, we're extracting fat. Oh, no, you forgot what you learned in analysis. You cannot extract fat from a big potato chip. You want to break it down, grind it into mesh 40, then extract your fat. So that's important. Okay, when you're preparing your samples, during preparation and during storage, you want to avoid certain things from happening, chemical changes, alterations. So enzyme elimination or control, why do we do that and how do we do that? Let's say your sample is high in lipase. What happens if lipase is active during your sample preparation or during storage? Zach? So lipase will break down your fat. So when you break down your fat, you're producing fatty acids, and then these fatty acids might react, might oxidize. Then if you are measuring rancidity, but your sample originally was not rancid, but you prepared it wrong and stored it wrong in a way that you enhanced breakdown by enzyme, you'll end up with wrong decision. You will make decision that your product is rancid when it really wasn't. So how do we eliminate it? Yeah, not Megan, sorry, Kim? Yeah, yes, freeze it. So that is most common, is freezing. Another way of eliminating an enzyme is inactivation by heat. But oftentimes you don't want to do that because heat will cause other changes. Depending on what you're measuring, heating will make you lose moisture, might denature your protein, might cause different changes that you don't want. So you could inactivate by heating, but oftentimes it is storing at very low temperature. -20 is a common freezing temperature that we store at. How do we avoid lipid oxidation? Which is, lipid oxidation can happen if you have lipoxygenase, let's say, if you have your sample is exposed to oxygen or light. So you can have autooxidation, photooxidation, or enzyme oxidation. So how do we avoid that? You don't want your sample to get oxidized, obviously. So how do we avoid lipid oxidation? Were you going to say anything, Jacob? What, say that? Yeah, so you can actually vacuum seal or flush with nitrogen. That means flushing with nitrogen where nitrogen will displace oxygen. So you can do that. That's one thing. There are other things you can do. You can simply add antioxidants. And you can also store at low temperature. So what else am I missing something? Oh, you can store in the dark. So oftentimes your sample is put in a container and then you wrap with aluminum foil, or you can put in amber bottles, that means dark glass bottles. Yeah. Of course, we don't want microbial growth. Why don't we want microbial growth or contamination? What would microbes do to our food? Well, if they produce toxin, I don't really care. I'm not going to eat it. I'm analyzing it. So, yeah, it will throw off the results. How? Because microbes often produce exogenous enzymes, so they will secrete the enzymes, and you will end up having fermentation sort of reaction, breakdown of lipids, breakdowns of fat, proteins, and sugars and carbohydrate. So the microbe's going to eat up your food and you end up having different results. So how do we avoid that? Megan, right? Okay. Yes, you can lower the pH. If the pH is not going to impact any other analysis, then you can do that. What else can you do, Axel?
Decrease the water activity.
You could. You can add salt. Again, you want any changes you make, you want to make sure that the testing you're going to do is not going to be impacted. So you can change the water activity. You can dry your product, or you can. Oftentimes we do dry it and get the moisture content, and then calculate based on dry bases and wet bases. So this is a common practice as well. Anything else we can do, Alex? Yes, we can store at cool temperatures. Again, -20 is a common thing. Another thing you can add is antimicrobial agents to prevent microbial growth. So whatever you do when you prepare your samples, during preparation and during storage, you want to avoid sample alteration. That is the big kind of message here. So this question is a study question in your book chapter. I don't know if it's still question number 7. This could be a carry-on from a previous edition. But what I would like you to do for a +5 opportunity, +5 points, is to answer this question and bring in that sheet to class on Monday for extra credit. You don't have to do it, but I encourage you to do it for extra credit. And I will post the key after Monday. On Wednesday, probably I'll post the key up so that you'll have an idea what the answer is. Okay, so I don't know if your copy has these questions. Does it have the recap question? Perfect. So again, review the recap question as you study, just to make sure you got the main idea of this lecture. And there are 4 more minutes. Last time I started a new topic, but because it's Friday, I'm going to let you go. I'll start new topic on Monday. Happy Friday.